are going to explore enzymes, the tiny protein molecules that are essential for all life. They act as biological catalysts, speeding up chemical reactions in our cells. Let's see what that means in more detail. First up, what are enzymes? Enzymes are protein molecules that act as biological catalysts. This means they speed up chemical reactions by lowering activation energy, the minimum energy required for a reaction to occur. All enzymes are made up of chains of amino acids linked by peptide bonds. They are found in every cells and are essential for metabolism, which includes breaking down molecules or catabolism and building them up or anabolism. Essential enzymes bind to specific molecules called substrates, convert them into products, and then remain unchanged so they can be used again and again. If you check out figure 3.1, you will see a diagram of an enzymatic reaction in action. Next, let's talk about the physical properties of enzymes. These include denaturation, which means when enzymes lose their shape, particularly their active site because of heat, extreme pH, heavy metals, high salt concentration. The second one is solubility. Enzymes need to dissolve in water or other solutions to function properly. The third one is colloidal nature. Enzymes are usually large molecules that do not easily pass through semi-permeable membranes. The fourth one is biocatalytic efficiency. A tiny amount of enzyme can convert a large quantity of substrate into products. Precipitation and molecular weight. We often use these properties to separate enzymes in the lab. The sixth one is enzyme activity. This is how fast an enzyme works, and it depends on factors like temperature, pH, and the concentration of both enzymes and substrate. See figure 3.2 for a visual summary of these physical properties. Enzymes also have important chemical properties. They are sensitive to heat and pH, meaning they only work well under optimal conditions. Their activity can be regulated by activator and inhibitor molecules. Enzymes are incredibly efficient catalysts that they can make reactions to occur up to 1,000,000 times faster than they would without them. They're also reversible, meaning they can help in both building up and breaking down molecules. And of course, enzymes are highly specific. They only work on particular substrates due to their unique active sites. Figure 3.3 highlights these chemical properties. The other one is what do enzymes do? Their main function is to speed up chemical reactions that are necessary for life. They play critical roles in processes like respiration, digestion, muscle and nerve function, and even in signaling for disease. For example, sucrase breaks down sucrose. Lactase breaks down lactose, which is found in milk. Protease helps break down proteins, and lipase breaks down fats. So figure 3.4 shows the steps by which enzymes act on substrates, making this reaction much faster. Enzymes work by lowering activation energy needed for reactions to occur. When substrate binds to an enzyme's active site, the enzyme helps convert it into a product. Then the product is released and the enzyme is ready to bind with another substrate. This process is illustrated in figure 3.5, where you can see how a lower activation energy leads to a faster reaction. Since enzymes are proteins, let's quickly review protein structures. Proteins have 4 levels of structure. The first one is primary structure, the linear sequence of amino acids. The second one is secondary structure, which is local folding into patterns like alpha helix and beta sheets held together by hydrogen bonds. The third one is tertiary structure, the overall three-dimensional shape of a single protein molecule. The fourth one is quaternary structure, the assembly of multiple protein subunits into a larger complex hemoglobin. Figures 3.636 through 3.9 and table 3.2 gives us overview of structural levels. Now let's talk about how enzymes interact with their substrates. There are two models to explain this binding. The first one is the lock and key model, and the second one is the induced fit model. So the lock and key model suggests that enzymes active site is a perfect fit for the substrate, just like key fits into lock. The induced fit models proposes that the active site changes shape slightly when the substrate binds, enhancing the enzyme's ability to catalyze the reaction. Refer to figure 3.11. Both models help us understand how enzymes are so specific and efficient. Enzyme activity is not constant, it is tightly regulated. There are several ways this happens. Allosteric enzymes. These have additional binding sites or allosteric sites where molecules can bind and change the enzyme shapes and activity, or figure 3.313. Genetic and covalent modification. Enzymes can be modified such as by phosphorylation, which adds a phosphate group and changes the enzyme's activity. Enzyme inhibition. This is when molecules called inhibitors reduce enzyme activity. There are reversible inhibitors, like competitive inhibitors that blocks the active site, and non-competitive inhibitors that bind elsewhere, and irreversible inhibitors that permanently inactivate the enzyme, which is shown in figure 3.15. And enzymes can be classified in a couple of ways. First, by their structure. The first one is simple proteins, which have active enzymes versus conjugated proteins, which is hollow enzymes. Hollow enzymes, which consists of an active protein part called an apoenzyme plus a non-proton part called cofactor. The cofactor can be a tightly bound metal or prosthetic group, or a loosely bound vitamin complex, which is known as coenzyme, as shown in figure 3.17. Enzymes are also categorized by the types of reaction they catalyze, such as oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligases. The names of most enzymes end in ASCE. Several factors affect enzyme activity. Temperature. Each enzyme has an optimal temperature, too high or too low, and the enzyme can denature, losing its shape. pH. Primarily, enzymes work best at optimal pH, and the deviation can impair functions. Figure 3.2 shows this one, and substrate concentration. The reaction rate increases with more substrate up to a point. Other factors, these include inhibitors, activators, radiation, water, and even feedback inhibition, where the end product of a reaction binds to an enzyme to slow down its activity. Figure 3.18 and 3.21 shows these effects. Enzyme kinetics is the study of how fast enzyme catalyzed reaction occur. One of the most well-known model is the Michels-Menten equation. Enzymes have vast applications across industries. They are used in food processing, feed, textiles, paper making, laser production, detergents, pharmaceuticals, and even molecular biology. For example, cellulose are used in biofuel production. Protease, amylase, and lipids are key in detergents and food processing. Rennin helps in cheese production, and the DNA polymerase and ligase are essential for genetic engineering. Table 3.3 provides a detailed look at these applications. Now let's shift gears and look at how enzymes are used in traditional practices in Ethiopia, specifically in malting. Malting is the process of converting grains into malt through controlled germination. This process involves steeping, which is soaking the grains in water to activate enzymes. The second one is germinating, allowing the grains to sprout and which modifies starches and proteins. Second one is cleaning. Cleaning, which refers to heating and germinated grain to dry it and develop flavors. The main purpose of malting is to produce enzymes such as alpha amylase and beta amylase, which breaks down starch into sugars. These sugars are then used by yeasts during fermentation to produce alcohol, as in the traditional Ethiopian beer called Allah. Figure points Figure 3.23 shows malted barley, which is commonly used in this process. To sum up, today we learned that enzymes are essential protein catalysts that speeds up chemical metabolic reactions by lowering activation energy. They have unique physical and chemical properties that affect their functions. Their actions are explained through models like the lock and key and induced fit models, and they are tightly regulated by various mechanisms. Enzymes are classified by both structure and types of reactions they catalyze. Finally, enzymes have diverse application in modern industries as well as in traditional processes like malting for local alcohol production in Ethiopia. Before we wrap up, here are some questions for you to think about. What are the key differences between the physical and the chemical properties of enzymes? How do the lock and the key induced Phi models help us understand enzyme substrate interactions? Which factors most affect activity and why? How are enzymes applied in both modern industries and traditional practices like malting? Feel free to leave your thoughts and answers in the comment below. Uh, that's it for today's deep dive into enzymes. I hope this video helped you understand how these powerful proteins work for their incredible role in both biology and industry. If you enjoyed this video, please hit the like button and share it with your friends and subscribe to Blackpill for more engaging science content. Thanks for watching, and as always, keep exploring and stay curious.